Now that you have chosen and validated your primary antibody, you are ready to proceed to the primary antibody incubation step, which takes place after the blocking step has been completed. First, you will need to dilute your chosen antibody in fresh blocking solution to achieve the concentration recommended by the antibody vendor for Western blot applications, making sure that the blocking solution is taken from the same stock that was used for blocking the membrane. You should also ensure that you now add tween 20 detergent to a final concentration of 0.2% to increase antibody specificity. Using too little detergent could lead to non-specific interactions, whereas using too much detergent could prevent specific antibody binding. The concentration of detergent can be optimized, but 0.2% is the recommended place to start. For the purposes of consistency, it's recommended to use a commercially available antibody diluent, such as Intercept T20 antibody diluent, which is available from LICOR. If you are multiplexing your detection, or in other words detecting two different targets at the same time, remember that you will need to use primary antibodies from different host species for targets that have the same or similar molecular weight, but avoid combining mouse and rat antibodies, as these have a high species similarity. These different primary antibodies can subsequently be independently detected by secondary antibodies specific to those primary antibody species and labelled with different fluorophores. If your targets are distinguishable by a differing molecular weight, you have the option to either use the same or different host species for the primary antibodies, depending on whether you would like them to be detected in the same or separate detection channels. In either case, however, you will first need to check for cross-reactivity between your primary antibodies to ensure that this does not affect your results. If you have two primary antibodies, A and B, a simple test is to run a couple of samples in triplicate, with a visible molecular weight marker in between. You can then cut the blot vertically in 3, following the lines of the marker lanes. Next, incubate one part with primary A only, one part with primary B only, and the last part with A and B together. Following secondary antibody incubation and detection, the resulting bands should have the same signal intensities in all cases, if cross-reactivity is not an issue. If it is, you will need to choose different primary antibodies for multiplexing or optimize your assay conditions. Once the primary antibody solution has been prepared, it can be used to incubate the membrane with gentle agitation for about 1 to 4 hours at room temperature, or overnight at 4 degrees Celsius. Once you've established your incubation time, you should stick to it for all future blots to ensure consistency in your data. Once the incubation is done, the next step is to wash the blot to remove any unbound antibody. First, you should pour off the antibody solution and then add some wash buffer, which will consist of either TBS or PBS with 0.1% tween 20. Make sure to pour this to the side of the blot and not directly on top of it. Quickly agitate the membrane for 10 seconds and then pour off the wash buffer. This step ensures that any residual unbound antibody is removed from the container. Next, you should wash the membrane for 4 times at room temperature for 5 minutes, and no more, always with gentle agitation every time. Be sure to strictly adhere to your set wash times every time to ensure consistency in your data. Now that we've gone through the steps required for the primary antibody incubation and for the washing of your Western blot, let's hand over to my colleague Jessica, who will go through some tips and best practices for achieving the highest quality results.